Hi, good afternoon. How you doing? This is chapter 6. If you are studying for exam 1, you may want to put this off for a little bit. We'll talk about it and we're going to add it into chapter 7 and 8. Chapter 7 and 8 are fairly short and I guess a little bit more fun. I don't know. I'm having trouble telling what fun is, but I think they're a little bit more fun. So practice the exam and then we'll get back to chapter 6. If you want to close this now, come back in a little bit. Otherwise, if you're going to continue on, I want you to consider some things that will be helpful as you go through chapter 6. The differences between prokaryotes and eukaryotic nucleus and DNA and nucleoid. Where do bacteria keep their genes and how many chromosomes do bacteria have? Bacteria only have one chromosome. So if that's true, how many alleles per gene do they have? Well, they're all haploid because they only have one chromosome, so they only have one allele for every gene, and that affects how they express. They don't have a backup gene. If something goes wrong with the one allele they have, they don't have a backup or another set of information that they can use to repair or deal with that. They're kind of stuck with the mistake. The other thing that this lecture is really going to focus on is where do bacteria get their diversity if they can't do meiosis? They can't do sex, they can't have meiosis, they don't do any recombination as far as we know yet. How do I end up with billions of kinds of bacteria then? So we're going to talk about the different genetic diversity and recombination because obviously bacteria are fairly diverse or we wouldn't have so many kinds. I do want to talk about some lab procedures that are coming up. So this lab is the PGLOW lab that we'll start with. You have a little bit of time. It's after the week of the exam. And I will, of course, do it with you. So it's not the only time you're going to hear this. And then we're going to start looking at horizontal versus vertical gene transfer. So we know that bacteria go through a process called binary fission. I make one bacteria when it's happy and everything's going well. It'll go through a S phase of interphase, essentially. It'll copy its DNA, and then it pulls itself in half. It goes through binary fission to dividing. And when it makes those babies are identical clones of the original parent cell. This should make it super easy for me to grow lots of homogeneous cultures of bacteria. All I need is to bring one bacteria to the lab. I put it on some sort of medium where there is food available for it, keep it happy, and after a while, no time at all, I should have a billion bacteria. But when I do that, in the real world, I don't get a billion identical bacteria. I get variation in it. Now, sort of as a side note, we know that bacteria are susceptible to some viruses. The viruses that they're susceptible to, particularly E. coli, is called a bacteriophage or a phage. And the one we're going to look at here, its name is the T1 phage. E. coli is particularly susceptible to the T1 phage. When you get these phages in your cultures of E. coli, it absolutely wipes them out. Almost all the E. coli die. Almost. Which means some of them survive. Now, for a very long time in our understanding of bacteria, we thought that this was the main mechanism of how bacteria adapted over time and became different. It's called the adaptation hypothesis. You may recognize it from other courses and biology that you took. The idea that the survivors get to pass their genes on and the ones that die don't. And over time, that will cause the genetic diversity in the population to change. We'll have a frequency of those alleles. The favorable ones will become more frequent. The less favorable ones will become less frequent. And 100%, this is something that happens. It's just not fast enough to explain how complicatedly diverse bacteria are. Urey and Delbruck suggested that bacteria, too, also go under spontaneous mutations, that they could just change their DNA. They're affected by sunlight, they're affected by the environment around them, and mutations randomly arise. This is something you've also taken in other classes. One of the mechanisms that drives evolution is mutations. Mutations bring new alleles into the population, and then if those alleles are favorable, they get passed into the offspring, and the offspring survive better. But again, it's not enough variation to explain the complexity of bacteria. Bacteria have only been around a couple billion years, not 100 million, a billion, 2 or 3, maybe 4 billion years at the most. They're too varied to explain waiting for adaptation and spontaneous mutations. So what we do, and we're going to show this and practice this in lab, is if I want to isolate mutations and understand them, I can do what's called a fluctuation test. Spontaneously mutated bacteria cells can actually be isolated. The ones we'll do and start with in lab are easy because they'll glow in the dark. So we have these glowing, it's called the P-Glow lab. We're going to end up with some bacteria that will glow. Now all we have to do is pick those bacteria out and plate them and then grow lots of glowing baby bacteria. Because bacteria haploid, if they have a mutation, they must pass it on. What we're going to do in lab is we're going to inoculate some fluid medium, so something in a test tube, and we're going to grow that in a culture or a whole mess of bacteria. One of the things I want to introduce here, but we'll talk about in lab, is bacteria can grow exponentially after their initial lag phase. What's going to happen is we're going to take one bacteria, and we're going to put it in the medium. It takes a little while for this bacteria to be happy and start to go through binary fission. But as it does, and it starts to go through doubling during binary fission, because it's growing at the same rate every 20 minutes or so, that's minutes, 20 minutes, what will happen is the population of bacteria will start to grow exponentially. And we'll see the log phase start where our bacteria colonies are growing very quickly. There is a maximum once they fill up the plate or they're using up all the nutrients, that colony will reach a stationary phase where the number of new bacteria is replacing the number of dying bacteria. After this, if you took microbiology, you know this starts to die off and the population starts to die as it uses up all the nutrients. The doubling time is how fast does it take each binary fission to happen and the population doubles. Some bacteria 20 minutes, some 90, some a day. We're gonna grow things that double very quickly. We grow them in a liquid medium, so that's going to be like a test tube. Here's over here. We're going to culture them until they grow this little cloud inside. And then you're going to take this and transfer them to an agar. An agar is a substrate. It has structure to it. And then we put nutrients in the agar, and that allows the bacteria to grow. So we provide them food and structure, and that makes them happy. Your goal is then to count how many colonies you get of your glowing bacteria. Now, here's the trick. You're going to plate and it's going to look like that when it grows. That's super hard to count. You want something more like this? That's a lot easier to count. So what we're going to do is a series of serial dilutions. We're going to take this and dilute it a little bit less, and then take that one and dilute it. We will work together in the lab to do this. And then for each one of these, you're going to take a sample and grow it out on an agar plate. Once they've had 24 hours or so to grow, you're going to select the dish that you can count the easiest. I choose this one. There's only 12 colonies on that one. These are colonies 1, 2, 3, 4. You can count if you don't believe me. There's 12. There's 2 up here that are hiding. These are the result of one bacteria that got put onto the agar, and it's had a chance to be happy and go through binary fission and grown a little colony, a group of identical clones. I want to ask you a question later that is, if I have these 12 colonies, what was my original concentration of bacteria? super easy to calculate. I'm not being facetious here. It's super easy to calculate. I have 12 colonies on my countable plate, and my countable plate was diluted 10 to the negative fifth. I take the number of colonies, time the dilutions, and that'll give me the number of original bacteria per milliliter in my original inoculum. We're going to take this and start talking about some genetic recombination. In bacteria, mutations and adaptation is not enough to explain the complexity of what we see in the real world. The hypothesis that has been kind of widely accepted about this is that now the bacteria are able to transfer genes from one living bacteria to another living bacteria. And there are a couple ways that it does that, but in general, this is called horizontal gene transfer. Today, it's going to be all bacteria doing this. In later lectures, I will show you how some eukaryotes have also participated in horizontal gene transfer. Normally, we talk about vertical gene transfer. That's from parent to child. That's how you got your chromosome set from mom and your chromosome set from your dad. But there are also ways of passing your genes onto someone who is still alive and existing, who may not be that closely related to you, especially if you're a bacteria. If you go through this and you're like, wow, this is awesome, and I agree, it's awesome. This paper down here, the Uprooting the Tree of Life, talks about horizontal and vertical gene transfer and its effect on the evolutionary pathway of life on Earth, and how bacteria particularly are affected by horizontal gene transfer. But if you look at this tree of life right here, you can see there's a lot of lines going horizontally, even into modern eukarya. Even the modern eukaryotes have benefited from some horizontal gene transfer in our ancestors. In bacteria, I've got 3 versions to talk to you about that allow for bacteria to go through genetic recombination, recombining their genes, without sex or meiosis playing a role, or fertilization playing a role in that. There are 3 things I want to talk about, and I want to talk about them in 2 ways. I want to talk about how they make bacteria more complicated, and how we use that to do some genetic analysis and understand the order of genes. We're going to talk about conjugation, mostly, but then also transformation and transduction. I did change the order of this a little bit from your textbook, just because in my mind it makes better sense the way I put it. So I want to introduce you to another experiment, and this one is in your homework. So I want to show you how to do it too. Once you do it 2 or 3 times, I think it actually makes a lot of sense. It becomes useful later. I'm hoping, ask me if this doesn't make sense before you do the homework. I have 2 different strains, so the same species, but different genes, alleles, that these 2 different strains of bacteria have. This is a list of the alleles that we care about for this experiment. And so this is Lederberg's and Tatum's experiment showing that bacteria can experience horizontal gene transfer. I grow in vial A, I grow an auxotrophic strain. Auxotrophic means that it's a mutant that is unable to synthesize all of its own nutrition. This one is unable to synthesize the first 2 compounds here. It says MET negative, BIO negative. That means it does not have a functioning allele to make those 2 things that it needs in its diet. It's going to need to get them from outside. It's going to need to feed on that in my nutrient source. Strain B here is very similar to strain A. It's also an auxotrophic mutation, but it has the ability to synthesize the first 2 nutritional requirements. It cannot synthesize the next 3. If I put strain A or strain B on what's called a minimal medium, or a surface and that provides no nutrition, they have to make it all themselves, neither of them survive because strain A is missing the ability to synthesize the first 2, and strain B is missing the ability to synthesize the last 3. If I put them together and I let them spend a little bit of time hanging out, if they work the way we thought bacteria worked, so no horizontal gene transfer, I would expect to have these two coexist and hang out together. But when I put them on a minimal medium, nobody should grow. Because strain A is still missing the ability to synthesize these two, and strain B is still missing the ability to synthesize these three. Everybody still can't make what they need to live. They need to get it from their medium. And so they should all be dead. But that's not what I see. When Lederberg and Tatum ran this experiment, so this is a mix, when they plated the bacteria on the minimal medium, some of them grew. Meaning that these guys, some of them, and it was very, very, very few of them, almost none of the original colony, but these few little colonies actually have the ability to synthesize all 5 of these nutritional requirements, even though none of the things I put in the original medium could do that. What they suspected was happening is the bacteria were swapping genes. One of the ways they can do that is through conjugation. This is bacterial sex. It's in quotes. It's not really sex, but it kind of feels like it when I describe it. It's a type of horizontal gene transfer where one of the bacteria actually has a gene that allows it to grow a tube and connect to the other bacteria. They can share cytoplasm, or cytosol, but more importantly, genes can be passed through that tube. In Tatum and Lederberg's experiment, it's unlikely that there was a spontaneous mutation that allowed all 3 of those alleles or all 2 of those alleles for one of the possible versions of the bacteria in the experiment to survive on minimal medium. So they set up what's called, this is a U-tube experiment, not like what you're watching this on, but it's a tube that shapes like a U. And in the middle of it, I put a filter. This is a filter. I put on the right side, I put a strain that is missing the ability to make nutrition. On the other side, I put strain A. It's missing a different kind of nutrition. But we're going to add one last thing. Some of the alleles, some of the bacteria in the strain A are going to possess an allele called the fertility allele. Your book calls it the fertility factor. If you have this fertility factor, you're able to go through bacterial conjugation. If I don't allow A and B to come into physical contact, I keep this strain here, nobody grows. They don't mix. I end up with everybody dying on the minimal medium. The bacteria here, this is a bacterium, the F+ allele causes it to grow a tube. I can't erase, but it's connected here, so this tube. And it grows towards the bacteria that does not have the F allele until they join. This is literally a physical process, it turns out. If I prevent this little tube, it's called a pilus, that's an I-L-U-S, multiple ones are called pili. If I allow this tube to grow and touch the second bacteria, they'll share genes. If I put a physical blockade in between them, I only have one color here, so it's kind of stinky. That pilus cannot pass through it. We will not transfer genes from one bacteria to the other. The F positive allele is not in the chromosome of the bacteria that carries it. It is an extra nucleoid piece of DNA. Your F+, your regular bacterium here has its nucleoid. This is its nucleoid. And then there it has this little ring of DNA hanging out all by itself. This little ring of DNA is called a plasmid. And in bacteria, we can find 1, 2, or sometimes 3 But it's not an infinite option to keep adding more plasmids on. They tend to compete with each other. Somebody always asks at this point when I'm doing this in class, if I can add DNA, how much DNA can I add? Not much. But a plasmid represents an allele, and all of the things that allele needs to be used. So it has a region so that it can be copied. It has a region so that it can be transcribed and translated. It can be turned into a protein. What this plasmid controls for, this F factor in a F positive cell, is this F factor controls for growing this tube. This tube, when it grows, joins a cell that has the F plasmid to a cell that does not have the F plasmid. That cell then, and this F factor is copied. It's passed into the receptor cell. It'll make a copy of itself and pass a copy in. This new cell that at one point over here did not have the F allele then gets a copy of the F allele, and now both of these are F positive cells. So it's kind of more like an infection than sex. There's a hypothesis actually that hypothesizes that this is the origin of viruses. And this is a protein tube that grows between two cells, and it has its own code for making its own copy of itself. It's kind of like a virus, right? And if you break this tube off halfway through when the plasmid is still inside of it, it's a virus. So to kind of think about it in two different ways, it's a way of transferring one gene from one existing bacteria to another existing bacteria. The recipient is now different than when this process started. It's got new genes. It's now a different kind of cell, but this is also a source of diversity in the bacterial population. In 1953, William Hayes actually isolated one of these F positive cells that seemed to be really, really good at this. It had a really high chance of recombining and spreading its genes and causing recombinant bacteria at the end of this story. He called it an HFR, or a high frequency recombiner. High frequency recombining is a slightly different story. This is the same picture. I'm going to change it in a little bit. The HFR strain, the fertility allele is instead of being over here as a plasmid, it's actually already incorporated into the gene of the bacterium. Sometimes I find the F plus code as a plasmid, but in high frequency recombiners, I find that gene has been inserted into the bacterial chromosome. So when it plays the code to grow the pilus and then spread itself into the receptor here, it drags the entire bacterial genome copy with it. So in the case of the receiving cell, it doesn't just get the F positive allele, it gets the F positive allele and then a version of some of the genes from the original bacterial chromosome along with the F plus allele. As that new HFR gene, so the factor and whatever it picked up from the original chromosome, moves into the cell that is receiving that, the cell, just for a minute or 10 minutes, becomes kind of diploid. It's not really diploid, but it gets two copies. It has two copies of every allele that was on its haploid gene, and that's not sustainable. But that moment where it becomes two copies, one from the donor and one that it already had in the recipient of every single allele, we call that cell a merozygote. It's kind of like a zygote, the result of fertilization, but it's not. It's not. It's not a zygote. The merozygote exists for a short period of time. It is a temporary state where the bacteria has received a second set of a copy of each of its allele, as as it ex- as it has those two alleles, it's no longer haploid. It's diploid for everything that got dragged into its cytosol. So how do we? That's great, right? You're like, wow, cool. It is, and it's super cool, right? But how does that actually help us? Well, we can map this. So those alleles in bacteria generally express. So unlike in eukaryotic genomes where you either express or don't express, or you maybe just never use that gene, or you only use it in some cells or certain stages of life, bacteria generally express all the time every gene on their genome. We'll talk a little bit later maybe about, switches to turn things on and off, but generally they express their genes. This process of dragging during a high frequency recombiner event of conjugation, this process takes time. It's not a instantaneous thing. It's something that takes minutes or hours of time. So if we allow them to conjugate and we let a high frequency recombiner combine with a cell that is negative for the F factor, the fertility allele, I can actually control the timing, stop the process at various intervals, and see what got dragged over. It's called the interrupted mating technique. It sounds rude, but it absolutely allows us to map the genome. We allow them to conjugate. We put them in a blender, for real. The blender breaks the pilus, so it separates the donor and the recipient. Because genes are transferred in an order from starting from where the high frequency fertility factor was inserted into the original genome, it will drag the genes and copy the genes from the original chromosome in the order they occur. They don't just randomly show up. They show up sort of like a ticker tape in a line. The ones that show up really early or get copied really early into the recipient were right next to where that gene for fertility was. Ones that take longer were further away from that gene for fertility. The ones that take the longest are going to be all the way back around to the beginning of that gene for fertility because it's in a circle. This is from your text. I'm going to show you this in a couple different ways. And then I have some practice in that homework for you about this. The practice in the homework is rough when you first look at it. It has a button there that says hint. If you look at it and you're like, holy cow, what? Click the hint. As you walk through the hints, and there's several, it actually walks you through a matching question that matches it that I think is very helpful. I think it will also help you think about how those 3-point mapping problems worked, so that maybe give you 2 bangs for your buck. I set up the homework so you get 2 attempts this time. So if you mess it up completely and miss all those points, you can go back and redo it. So I want you to get this idea down. But the idea is I can use time and controlling their mating and interrupting their mating to figure out the order of genes. I allow lots of conjugation and lots of interruptions. And then I basically do time mapping. I allow them to move for 1 minute across that conjugation tube, across the pilus. And then I put them in a blender. That will have dragged the gene for fertility and whatever was right next to it. Then I allow them to mate for 2 minutes. When I allow them to mate for 2 minutes, that will drag the high frequency gene, the first gene next to it, and then the gene that was right behind the first gene next to it. I can start to tell, because then the recipient will express those genes, I can start to tell what order those genes exist on that bacterial chromosome. Now we have several strains of high frequency recombiners. We determine the strains, the varieties of them, based on the order of those nutritional genes. And so kind of as a side note, the ones that bacteria are all about eating and reproducing and surviving. A lot of the genes we are mapping here at these examples are nutritional nutritional genes. We take some bacteria that are unable to make the nutrients they need. We can map out, we can let them conjugate and pass those on, and then we put them on a minimal medium, and if they can't survive, we can see the expression or not expression of those genes. I am going to, in the homework, it walks you through how to figure out these genes. I will show you I will probably do it not the day of the exam, but the week after. I'll have some practice questions as we go through setting up and doing some of the prep for this PGLOW exam or lab as well on how to do this. You're looking for the order of the genes and their relationship to the HFRF positive allele. Come back to that in the homework. We'll come back and talk about that in lecture. I don't have any practice questions here because I want you to walk through the one in the homework for chapter 6. The chapter 6 homework is half work through questions and half multiple choice practice kind of questions. So some of them should be simple. Some of them should not be. And then again, always ask me, send me an email if you're not sure about what you're doing. And I'll see you in the next video. We'll continue on and looking at some other ways of bacterial recombination. All right, I'll see you soon. Bye-bye.